evening, students. Today's topic is immune system. In this pandemic situation, we often come across the term immunity. Everyone is saying that we must improve our immunity to fight with coronavirus. What is this immunity? What are the different components? What are the different types? Everything we'll discuss in detail. First, let us see what is immunity. The overall ability of an individual to fight against the disease-causing germs or organisms is immunity. That means if any pathogen enters the body, there is a system that fights with that particular antigen. This is nothing but the immunity. And the different components that are involved in immunity together called as immune system. What are the different components? Organs, cells, etc., which bring up the immunity is called immune system. And the study of immune system is called immunology. Father of immunology, Edward Jenner. Then if we look at the types of immunity based on the nature of response. Based on the nature of response, the immunity can be broadly classified into two types: innate immunity and acquired immunity. Let us see the difference between innate and acquired. This innate is inborn. That means it is seen at the time of birth. And this develops during growth of the animal. And this is the same in all the individuals of a species. It is different in different individuals of the same species. It does not provide immunological memory. Memory is absent, but it has a memory. Once the antigen enters the body, it can recognize it. If the same antigen enters the body second time, it can easily recognize the same antigen. That is the difference between innate and acquired. First, we'll say about the innate immunity. In this innate immunity, we can observe different type of barriers. Now, what are the different barriers which are found in this immune system? We'll see. Number one, physical barriers. This includes skin and a mucous membrane. You know that skin is the largest organ found in our body, and the skin does not allow the pathogens to move into the body. And next, mucous membrane. The mucous membrane lines the respiratory tract, GI tract, and urinogenital tract. Suppose if the pathogen try to move through the nose or mouth or through the anal route, whatever it may be, the mucous membrane consists of the immune system or the immune components, what we call it as a MALT, mucosa associated lymphoid tissue, whose percentage is nearly 50% of the total lymphoid tissue. And this mucous membrane can trap the pathogens and try to destroy the pathogen. So the physical barriers are skin and mucous membrane. Suppose if they cross the physical barriers, we can observe physiological barriers. Now, what are the physiological barriers? We can observe lysozyme found in saliva, tears, and also found in intestinal juice. They can kill the bacteria, especially these enzymes. This is lysozyme can kill the bacteria. And HCl found in stomach. And then acidic pH found in vagina. All these components that try to destroy the pathogens. So the skin mucous membrane physical barriers and lysozymes, HCl, pH of vagina, all these come under the physiological barriers. Now here the physical and physiological barriers together considered as a first line of defense. So physical, physiological barriers come under first line of defense. Next, coming to the other barriers. Next, cellular barriers. Now these cellular barriers include mononuclear phagocytes like monocytes. I think you know the monocytes transform into macrophages when they reach the different parts of the tissue. Number 2, polymorphonuclear phagocytes. Best example, neutrophils. So they fight with the infectious germs. And then NK cells, natural killer cells. They are a type of lymphocyte. All these come under cellular barriers. Usually the macrophages are found in tissues. If the pathogen reaches the tissues, they can engulf. That means they can kill the pathogen. And when the pathogen is found in blood, generally neutrophils can destroy. NK cells, natural killer cells. I'll explain the mechanism of the action of natural killer cells later. Next, cytokine barriers. Now here the cytokine barriers, interferons. Now these interferons are antiviral proteins which are generally produced by the virus infected cells. Now these antiviral proteins are generally released by the virus infected cell. They just enter the neighbor cells and bring antiviral state in the neighbor cells. So the interferons are generally called as antiviral proteins. Both cellular and cytokine barriers together considered as second line of defense. So skin, mucous membrane, both together considered as physical barriers, cytokine barriers, enzymes, HCl, etc. Both physical and physiological come under first line. Cellular and cytokine barriers come under second line. If first line fails, we can observe the action of second line. If second line fails, then you can observe the action of third line. Now, what are the components which are found in third line? B and T lymphocytes. Now, B and T lymphocytes, they are involved in third line of defense. And of course, this is also called as acquired. So first and second lines come under innate immunity and third line come under acquired immunity. Now before we go to the acquired immunity, Let us see the different lymphatic organs found in human body. Lymphatic organs found in human. Now, organs of immune system. Now, what are the different organs involved in immune system? Here, the organs are broadly classified into two types: primary lymphoid organs and secondary lymphoid organs. Now, primary lymphoid organs: bone marrow and thymus. Here, the secondary lymphoid organs: spleen and other lymph nodes. All come under secondary lymphoid organs. Now here, what are these organs? How they are involved in development of immunocompetence, we'll see one by one. Now, primary lymphoid organs are those where the B and T cells, they develop into antigen sensitive cells. That means they undergo maturation and now they are capable of recognizing the antigen. So B and T cells develop into antigen sensitive cells in primary lymphoid organs. Whereas in case of secondary lymphoid organs, we can observe the activation of immune cells like B and T cells. What happened in this particular area? So secondary lymphoid organs are those where antigens are trapped, so that whenever the B and T cells reaches the secondary lymphoid organs, there they can recognize the antigen, they become active. So let us discuss these primary lymphoid organs in detail. Now, primary lymphoid organs are bone marrow, thymus. Bone marrow is the place where we can observe the production of different type of form elements, that is blood cells. So within a red bone marrow, we can observe hemopoietic stem cell. I think you know stem cell is one which can able to give rise to different type of cells. A hemopoietic stem cell can give rise to two secondary stem cells like lymphoid progenitor and myeloid progenitor. Now, lymphoid progenitor is one who can able to give rise to three types of cells: B cells, T cells, and NK. That means NK is also a type of lymphocyte. On the other side, myeloid progenitor can give rise to the rest of the blood cells like RBC and rest of the WBC. So, whether it is B or T lymphocyte, their formation takes place in bone marrow. In case of B cell, there itself it undergo maturation, that is it develops into antigen sensitive cell. It develops surface antibodies and becomes antigen sensitive or mature cell. This is antigen sensitive or mature cell. And after the maturation, they enter the bloodstream. From there, They reaches the secondary lymphoid organs where they can recognize the antigen if present. So they become active B lymphocytes. On the other side, T cell formation takes place in bone marrow. After that, it leaves the bone marrow, reaches the thymus, where it develops into immunocompetent or mature or antigen sensitive cell. So this is the place where you can observe the differentiation. It can transform into TH, it means T helper cells, or TC, cytotoxic cells. So formation takes place in bone marrow, but the maturation takes place in thymus. You know that thymus is a lobed gland present below the breastbone. During development up to puberty, the size increases, and after that the size decreases. Because of this reason, in old people, the immunity power is less. And after the maturation, the same T cells reaches the secondary lymphoid organs where they can recognize the antigen and become same active cells. So this clearly indicates that primary lymphoid organs are those where you can observe the formation and maturation. So B lymphocytes represents bone marrow or bursa of birds, and T cell represents thymus. Now we'll see the secondary lymphoid organs. Now the secondary lymphoid organs includes spleen, lymph nodes, tonsils, malt. And here spleen is the largest lymphoid organ in which we can observe both the lymphocytes and phagocytes. Lymphocytes and phagocytes. Now generally, the blood generally passing through the spleen, it consists of antigens. They can be trapped by lymphocytes. If this blood is having old or damaged RBC, they can be destroyed by phagocytes, nothing but macrophages. On the other side, lymph nodes, tonsils, and malt, all these can trap the tissue fluid borne antigen. Suppose if any antigens found in the tissue fluid when enter the lymphatic system, when they are passing through the lymph nodes, they can be trapped by the lymphocytes. So here, the tissue-borne antigens can be recognized by, or tissue fluid-borne antigens can be recognized by the lymph nodes and other structures, whereas the blood-borne antigens can be recognized by spleen. So there's the formation, activation takes place in two different areas. The formation, maturation takes place in primary lymphoid organs, whereas the activation takes place in secondary lymphoid organs. So that's all for today. Tomorrow we'll discuss about the acquired immunity, B cells, T cells, everything. Thank you.
Thank you for watching the video. Please subscribe Dr. RK's classes for more important videos. Thank you.